In this video, we're going to conclude our overview of organic reactions with a quick look at organic compared to biological reactions. Now, as an organic chemist myself, my goal is not to convert all of you to organic chemists, although, if I convince a few of you, that'll be great. But I want you to keep studying biology and biochemistry and whatever it is that really interests you with an understanding and an appreciation of the relevance of organic chemistry. Because biochemistry, for example, is just organic chemistry happening in the body. As you learn more about different biochemical reactions, what's happening in the body, what's happening in different microorganisms, you're going to see the same types of transformations that we are learning about in organic chemistry, just in a different environment. So what are the differences, though, between organic reactions in the lab and organic reactions in biology? So one of the big differences is that many lab reactions are going to be carried out in a particular solvent, such as some organic liquid. We've worked with diethyl ether, we've worked with dichloromethane, so many reactions would be carried out in solvents such as these, whereas the biological reaction is going to occur in the aqueous environment that is in cells. In the lab, reactions might be carried out at very different temperatures. So some reactions, we need to cool them down a lot. Maybe we cool it down to -80 degrees. Some reactions, we have to heat up a lot to get them to proceed at a decent rate. And so we might have to warm it up to 150 degrees. But biological reactions, well, we can't just throw ourselves in a -80 freezer or heat ourselves up to 150 degrees. So biological reactions are limited to the temperature of an organism. So maybe we're talking about 37 degrees Celsius. For many organic reactions in the lab, no catalyst will be used, or maybe simple catalysts will be used, whereas for biological reactions, really large complex enzymes are needed. Enzymes are the biological catalysts, and this is why it's possible for a reaction to be carried out at 37 degrees when we might otherwise have to heat it up to 150. Here we have an energy diagram of some generic reaction. The pink line represents the uncatalyzed reaction pathway. So here's our reactant, here's our product. This would be our transition state up here. And so for this one, this would be our activation energy. We don't know our scale, but let's say that because this is such a big activation energy, this is why we might have to heat it up to 150 degrees. Now, when we talk about a catalyzed reaction, it's kind of tempting to say that instead of proceeding like this, then it proceeds like this. But actually, it probably proceeds in many, many steps. Here we have a step that brings us to an intermediate, another step, another intermediate, another step, another intermediate, another step to our product, for example. And in this case, we have an activation energy for step 1, an activation energy for step 2, an activation energy for step 3, and an activation energy for step 4. In each case, the starting point is the reactant of that step. These sizes are pretty similar. It looks like this one might be the largest activation energy, and so this one would be the rate determining step. But what I want you to notice is that this activation energy is much, much smaller than this activation energy. So bad news when it comes to biological catalysts is that it requires really large complex enzymes. But the good news is that reactions can be carried out at a particular temperature because the catalysts provide a pathway with a low activation energy. Finally, when we think about the specificity of a reaction or of a catalyzed reaction, there's usually very little specificity for the substrate in a lab reaction. And so we can use the same reagent on a bunch of different starting materials and we'll get the same type of product. Whereas in biological systems, the enzymes typically have very, very high specificity. And so a particular enzyme may catalyze a reaction only on a single substrate, and often it will do so with complete regioselectivity and stereoselectivity. So this concludes our overview and introduction to organic reactions and some of the characteristics and features of organic reactions. We are now ready to start digging into specific transformations.